Okay, so today I'm going to talk to you about meiosis. So meiosis is the division of your cells in your body, specifically either in the ovaries or in the testes. Okay, so if you're a female, meiosis only happens in the ovaries, and if you're a male, it only happens in the testes, because this division is going to give you one specific cell, and that's going to be a gamete. Okay, and your gamete is the biological term for your sex cells. So if you're female, those gametes, you've heard them called the egg cell, but we call them oocytes. And then for males, those are going to be sperm cells. So all other cells in your body are going to do cell division to make one cell divide into two cells through the process of mitosis. Okay, so if you cut yourself on your arm and you have to replace some of the cells, other skin cells in the vicinity will divide and make two new cells. So in a human, that number is 46 chromosomes in every single cell that you have. And in the end, you're going to end up with 46 chromosomes to replace the ones that have been dead or damaged, or maybe you're growing, whatever. Okay, mitosis, the definition of mitosis is division of the nucleus. And it has 4 steps, PMAT, which is prophase, metaphase, anaphase, and telophase. And that will be your mitotic stage. That is not mitosis. Mitosis. Okay, and then after that, you're going to get cytokinesis, which is division of the cytoplasm. So this one cell, the chromosomes will do everything they do. It looks kind of like a peanut. It starts pinching in, doing cytokinesis here, and then that's your cleavage. And then once it completely separates, then you're going to get two new cells. Now, we have numbers to delineate the number of chromosomes in these cells that I'm showing you. So a cell that has all your chromosomes in it is called a 2N. And 2N just means you have everything. You have all the 46, okay? And then in mitosis, each new cell is also going to be a 2N. So if you had an organism that had 30 was their 2N number, then the two new cells that they made, if they made them correctly, would also have 30 chromosomes in them. Meiosis is going to do a little bit differently, because meiosis is going to make the reproductive cells. And the reproductive cells have to end up coming together to make that first somatic cell. So if you do math backwards, that first somatic cell is a 2N. So for us, that means 46 chromosomes. So that means half had to come from the oocyte and half had to come from the sperm cell. So sperm cell has to have 23 chromosomes, and the oocyte egg cell has to have 23 chromosomes. Otherwise, 23 and 24 would not make 46, it would make 47. Or 46 and 46 would not make a 46, it would make a 92. And that would not make an organism. So gametogenesis is the term for making the gametes, and it turns a diploid cell into a haploid cell. And it actually doesn't do 4 steps, like mitosis is just 4 steps. Meiosis is going to be 8 steps, and they're going to have a very similar pattern. Okay, so I'm going to go to a new sheet. All right, so mitosis was 4 steps and with 2 new cells. Meiosis, we're going to start with a cell, and we're going to go through prophase 1, metaphase 1, anaphase 1, telophase 1. And we're going to end up with 2 cells once that first rotation is over. Then it's going to go prophase 2, metaphase 2, anaphase 2, telophase 2, okay? So that cell will go through all those stages and will divide in half, and also that cell goes through and divides in half. So after a full meiosis, okay, that's 8 steps, you go from 1 cell to 4. The first round is called meiosis 1, and the second round is called meiosis 2. Okay? And they're both PMAT, cytokinesis. PMAT, cytokinesis. But what happens in this first round of meiosis is we go from our beginning cell, and I'll talk about where that comes from in just a second, but your beginning cell in a human cell is going to have all the DNA available, possible, period, okay? So this cell, if it represents you, has every single chromosome that you have available. But at the end of this first division, it literally takes everything you have and it splits it in half. So it goes from a diploid, di meaning two, to haploid cells. This is so that you can actually give half of what you have to the baby and combine it with another partner somewhere along the way in the process of sexual reproduction to create a new organism that has 46 chromosomes. So in this area right here is where somehow these chromosomes line up and know to separate into two groups. And then the second division, it just makes an identical copy. Okay, so this is very similar to mitosis. So all the steps here look just like mitosis. Okay, so I'm going to write looks like mitosis. And that one cell will go through mitotic division and make two new identical haploid cells, and then this one goes through and makes two new cells. So these two are actually identical, and these two are actually identical, but they're different, they're complete opposites of each other. All right, so if you're a male, this is going to be in the testes. All right. And if you are a female, this is going to be in the ovary. Okay. So there's two testes and two ovaries. And the cells that start there are cells called germ cells. And it comes from the term germinate or germination because we're going to end up using these cells. They're like the seeds of a plant, and seeds of plants germinate. And so we talk about these cells being your germ cells. Because they end up germinating, making a cell that can turn into a human if it combines with another germ cell, or excuse me, another gamete. So here are your germ cells in your testes and your ovaries. Now, boys, you do not start this process until puberty. All right, so the hormones will kick in. They'll start telling the testes to start releasing testosterone, and testosterone is going to start making this production of the sperm cells. So the germ cells in the testes will start going through these phases. It will start making these cells that have 23 chromosomes. And then those cells will go through a series of tubes and passageways. And while it's going through those tubes and passageways, it will add a tail so that it can swim. It'll give it some mitochondria because these sperm cells are going to need energy to swim and move. And then it'll add enzymes to the front of they call this like the head of the sperm. So the sperm cell, like I said, you get the tail added. They add mitochondria here to help it grow. Help it move. And then at the tip of the sperm cell are enzymes. And these enzymes are specifically made to break through the barrier of the oocyte because his job is to hopefully deliver his 23 chromosomes to whatever egg he comes in contact with. Now, most sperm cells are going to not make it. Even just once they enter the female's body is so acidic and caustic and has her own enzymes because she's trying to kill off all the weak sperm. So most of the sperm cells that enter the body don't even live. And then the ones that do live, they have to deal with a lot of other pressures and challenges before they even get to the egg. So this is nature's way of ensuring that sperm cells that have wrong chromosomal numbers or their mitochondria are weak or whatever, they just don't make it there and they don't fertilize the egg. Okay, so now with the females, a little bit different stuff happens. We still have females going through meiosis, but the meiosis starts a little bit earlier for females. So I'm going to write it. I'll write it in purple. No, I won't write it in purple. I'll write it in blue. Okay, so for females, we do meiosis just as well, but we do this first phase of meiosis 1 during gestation. Okay, and during gestation means while we are actually developing inside of our mom. So as all of our body parts are developing, our ovaries are also developing, but the cells in the ovaries, which are germ cells, also for females, they start this process prior to birth. So they go through this first meiotic division, and then they stop. And so right here, they are arrested development is kind of the term for them. So when a female is born, she already has all her eggs have already gone through this first phase of meiosis. So then they only need to go through this second phase. All right. So for females, this is when puberty starts. Okay. When puberty starts, then one of these cells a month will go through meiosis. Dramatically different from males. All right, millions of times this occurs daily for sperm production in males. And this is all mammals, all male mammals that make sperm cells. It's just millions of them a day. The egg is so important for the viability of the offspring that the female can't risk making thousands and thousands and millions of them. They really focus on making one good egg every month. And that one good egg really has to be perfect, because the thing about the egg, the oocyte, it does have 23 chromosomes for mom in it, but it also has all the organelles that the baby will need. Dad is only going to be able to deliver the sperm cells and perhaps maybe a few mitochondria. But in general, mom has to give the baby everything it's going to need, including half of her DNA. So this production is super slow, and only one is focused on. So they don't focus on millions, they focus on one. So in this first meiotic division, in a female, they actually make one giant cell from the original germ cell. And then this little guy right here is so kind that he donates all of his cytoplasm to that one cell. So they're divided in half, but sperm cells would divide evenly. These egg cells, these oocytes, you get one and one like this. Okay, so this guy gives all his cytoplasm to one cell. So it's unequally distributed and divided. And that's so that this one cell literally is the one that gets released. So this tiny one to the side, we call a polar body. Now, that is somewhere in the ovaries. Sometimes it's reabsorbed by the ovaries and used and recycled to make other parts, or it's sent to other parts of the body for different things, but it's never meant to turn into an oocyte. This is what will go through that second phase of division when girls go through puberty, and will actually make the end result. So a female going through meiosis, the images look a little bit different. So we start with this cell this size with 46 chromosomes, and I'm in my ovary. All right, it goes through the first phase of meiosis, so P1, M1, A1, T1, and then cytokinesis. And then I'm going to get one that has 23 and one that has 23. Okay, so this is called a polar body. And typically, it doesn't go through the next round of divisions because there's really no point. This happens during gestation, so while the female is developing, and this happens during puberty. Okay, and puberty is going to give you that second round of meiosis, so P2, M2, A2, T2. Once again, you'll get a small with 23, and then you'll get a big one with 23. Okay, so this is also called a polar body. Now, if this guy decides to divide as well, then they'll still stay tiny and they're all polar bodies. And this one actually gets the term oocyte. Okay, so that oocyte, this process takes, this is all hormone driven. So hormones go up and down during this process. This would be prior to ovulation. When this oocyte gets released from the ovary, we call this ovulation. And she has a couple of days before she can get fertilized. And once she gets fertilized, let's say right here, we have a sperm cell that comes in and fertilizes, and we'll talk about all about the crazy stuff that happens here. Then she waits about a day. Some organisms, within a couple minutes, they start dividing again. Some organisms within a couple hours. Humans, I would say a safe bet is for about 24 hours. It kind of regroups, realizes, all right, now it's not just a 23 and a 23, but it's a cell. It's a diploid cell with 46 chromosomes. So it kind of hangs out. The chromosomes get matched up, pair up, and then within 24 hours, it'll divide into two new cells with 46 chromosomes. And then that division is mitosis. Okay, so mitosis is what continues to go on and on and on to grow our cute little baby that we're going to have right here. Wrap him in a little blanket. Okay, this is our little baby with his little passy. We'll let him go to sleep. Okay, so those babies that are born are coming from this meiosis that occurs to create our oocyte. Over here, I have my sperm coming in that was created also by meiosis. They join together in the process of fertilization. They stay as a single cell, maybe about a day or so, and then they go through their first mitotic division. They stay like this for maybe about a day, and then they start rapidly dividing, and they're going to go through a lot of other divisions. Okay, so I just want to go a little bit more in depth on some of the stages of meiosis. And what I've done is I've created a long sheet so I can keep flowing through the stages of meiosis. We're going to start, we start with a germ cell and has 46 chromosomes. And then when it gets the trigger from hormones, okay, and so this would be male hormones in this situation. For females, what I'm drawing, these first 4 steps will happen to you during gestation. But that cell in that ovary or that testy will, the nuclear membrane disappears. Okay, so I'm going to draw a little dotted line around my nuclear membrane. The chromosomes will become visible, and they'll be fully duplicated chromosomes. Okay, and if I was to draw every single one of them, I would have to draw 46 of them. Another thing that's going to occur is your centrioles are going to appear, and they're going to sometimes they draw them looking like shiny, because those shiny centrioles are what the spindle fibers are going to come out of. So these are your spindle fibers. And these are going to be responsible for moving and arranging the chromosomes along the equator so that everybody lines up like they should. All right, so I'm going to give a color to this pair. I'm going to actually outline them in green to indicate that these two are homologous chromosomes, okay? Homologous chromosomes. And that means that they both have genes for the same stuff on them. So we number chromosomes based on size. And so every human has 46 chromosomes, but you have 2 of them that are the same size, that have the same genes on them. One of them you got from your mom and one of them you got from your dad. So we call them homologous chromosomes, and they're in pairs. All right, when you do meiosis, you have a number 1 and a number 1. You'll give one to one cell and one to the other cell. You'll split them and separate them. This is the second set of chromosomes, et cetera, et cetera. Now, males, inside the chromosomes, you're going to have, these would be your sex chromosomes. So this is what they call them, X and Y, but it doesn't look like a Y. The Y chromosome is just really tiny. If this was females, this would be 2 big ones, but these are your sex chromosomes. So when males make sperm cells, half their sperm cells get an X and half their sperm cells get a Y. So there's literally a 50-50 chance that we're going to have a baby girl, 50-50 chance that we'll have a baby boy. And it's actually the father that determines it, because only the father has that Y chromosome. If the baby doesn't get the Y chromosome, then the baby's a girl. All right. So in this prophase 1 of meiosis, another crazy thing happens, and it's called crossing over. Okay, and crossing over occurs when your homologous chromosomes, and I'll zoom in on this pair. I'll draw one of them all green, and then I'll draw one of them outlined in green. Okay, so we'll say that this person got this chromosome 1 from their mom and this chromosome 1 from their dad. All right, and we'll say that on here is the gene for eye color. So from dad, you got the big B dominant brown-eyed gene, and from mom, you got the little B blue-eyed gene. All right, and so for some reason, chromosomes during prophase 1, only in meiosis, when they are going through prophase, they go and they find their partner and they bunch up. And sometimes when they bunch up, it causes pieces and parts of them to cross over and overlap. So if these guys were pairing up, they might look like this under the microscope. So they're trying to stay so close together so that they don't get separated in the next phase, because they want to line up in line together during metaphase. So when that happens, sometimes they exchange information, and scientists aren't really sure why. So this pair of chromosomes afterwards might end up looking like this. The fully dark green chromosome that this person got from their mom now has a little piece of DNA from their dad, and the uncolored chromosome here that they got from their dad it's a little bit of mixing and matching here. So these pieces just swapped places. So we call it crossing over. We didn't lose any information. Both of these pieces are exactly the same. It's just, this one, the blue-eyed gene came over, and that one, the brown-eyed gene came over. So they swap around and mix up. This adds to variety, which adds to a species' ability to adapt and evolve, because variety allows for all kind of new traits to show up. And sometimes the traits aren't that good, and so they go away. But sometimes it adds a new trait to the population that's actually very beneficial. All right, so all this is happening in prophase. So if you're asked, when does crossing over occur? It occurs in prophase 1, and it's just when homologous chromosomes exchange information, no mutations occur. It just increases the amount of variety that an organism can have. Okay, so then we'll go into metaphase 1. All right, and metaphase is always the lining up phase. And in mitosis, they lined up on the equator. They're going to do the same thing here. And on meiosis, they line up on the equator. Difference is, not in the centrioles, because their centrioles still head to the poles. Get their little spindle fibers out there. But these chromosomes are now going to line up in pairs along the equator. And it's very random. Like, it's not, they don't line up pair 2, pair 3, pair 4, etc. like that. There's going to be 23 pairs. They just definitely line up in pairs. And they don't line up with the chromosome from mom always on the left and the chromosome from dad always on the right. They mix and match. So if you were to sit down and take one person's chromosomes and pair up their chromosomes and line them up down the equator and figure out all the different combinations of who would be on the left and who would be on the right and how they would line up, one person could have 2 to the 23rd different arrangements of these pairs of chromosomes along this equator. That's why there's so much variety in this world, because there's so many different ways that humans and bears and dogs and cats and frogs and fishes and all those animals, it's so many different ways that they can create their gametes and so many different combinations. So there's a lot of uniqueness in organisms. So these chromosomes are lining up in pairs, single file down the middle, and then these spindles are going to attach, but they're only going to attach to their full chromosome, okay? So we're not ripping chromosomes in half like we did in mitosis. We literally want to take, and it's like we're dividing things out. Okay, you get a 1, you get a 1. You get a 2, you get a 2. You get a 3, you get a 3, et cetera, et cetera, et cetera, et cetera, et cetera, all the way down. So we get to that 23rd pair, and the 23rd pair You get this one and you get that one, okay? So that division phase is anaphase. We're going to call it anaphase 1. And in that division phase, the cell kind of starts looking more like a little watermelon because these centrioles are going to pull their full chromosome, not a chromatid this time, but this whole entire chromosome gets separated from its homologous partner. Okay, so that gets a whole chromosome and that gets a whole chromosome. Same thing here. Full chromosome, full chromosome. If this screws up, you get some messed up egg cells or sperm cells. So this has to happen. So we get 23 full chromosomes go into this new cell, and 23 full chromosomes go into this new cell. So we are going from a diploid germ cell, diploid, which means you have all the DNA, all the possible chromosomes, and it's called a 2N. And then we're turning it into 2 haploid cells, which have half. So we call those N. So they have half all the DNA of the original organism. All right, and then it'll go through telophase 1, and then it'll go through cytokinesis. And telophase 1, basically, this cell kind of starts pinching in. A new nuclear membrane starts to form so that we have 2 new nuclei forming. Inside of this new nucleus, we have full chromosomes, okay? And over here, we have our full chromosomes. And if we were doing all the numbers there were in a human, this would have 23 full chromosomes, and this would have 23 full chromosomes. This would have a 1, a 2, a 3, a 4, a 5, a 6, 7, 8, 9, 10, etc. And this would also have a 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, all the way to 23. And then in cytokinesis, we're going to end up with 2 completely separate cells with half of our parental DNA, so half of what we started with, going here. Okay, so this is kind of how genetics is going to work. And our next unit is going to be with Punnett squares, and this is how we're going to be able to figure out how to do Punnett squares. Okay, so that is our first round of meiosis, and it's called meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1, and then your cytokinesis, and you end up with 2 haploid cells. All right, girls, you've already done this for every cell in your ovaries. Boys, you didn't start doing this until you went through puberty. The second round is continuous for males. For females, this is where our puberty starts. Okay. So now, take my paper because I made it long. I'm continuing from this little spot right here. Okay. And we're going to go into that second round of division. All right. One little quick note before I go there. All right. Back at metaphase 1 and anaphase 1. If for some reason the chromosomes didn't separate equally and this chromosome accidentally comes over here with this one, then that's going to give this cell a chromosome number of not 23, but 24. And that's going to give this cell a chromosome number of 22. Not good, okay? We need it to be 23 and 23 or you're going to have problems. Now, if these are egg cells, then it's just not going to develop and you have issues. If these are sperm cells, it may continue, but it will never be able to make it to survive in the body because it'll die as soon as it gets there. This process of not separating like you should is called non-disjunction. And non-disjunction is what causes extra chromosomes or fewer than normal chromosomes. Okay, and there's a lot of disorders that we can talk about. Most common disorder caused by non-disjunction is Down syndrome, and that's where you get the 21s instead of a 21 going left and a 21 going right, 2 of them go in one spot. So that's not good. All right. So go back here to the second phase of meiosis. Pretty straightforward. We're going to go prophase 2. So each one of these new cells will move into nuclear membrane disappears, nuclear membrane disappears, the chromosomes become visible, and the centrioles show up. Literally no different from any of the other prophases. It's just our chromosome number is now different. Next, we transition into M2. Metaphase is the lining up phase. It's going to look just like mitosis. So both of these cells, the centrioles will head to the poles. The chromosomes will now line up single file because they do not have a sister. The spindles will come out and they will attach to half of the chromosome, just like mitosis, okay? Because what we're going to end up doing here is we're going to take this chromosome, which is actually two sisters joined together, and we're going to take that centromere and we're going to dissolve it with an enzyme. We're going to rip it in half. So anaphase 2 is when we pull the sister chromatids away from each other. So it starts kind of looking elongated. All right. So in anaphase 2, all of a sudden this centromere disappears due to an enzyme. So an enzyme breaks down the centromere. Okay, so that's the glue. When that glue is gone, then we can play tug of war, and then all of a sudden I get to rip the sister in half, okay? And this gets pulled that way rapidly. So we're just tugging it back towards these two new nuclei. These centrioles, I'm gonna rip these in half and pull them this way, okay? So we should know by now, if you don't know by now, these are identical to each other. So all you need is half of this. Now, when it is together like this, the whole thing is called a chromosome and each half is called a sister chromatid. But when you rip it in half and it is independent and not attached, they now actually call this whole thing a chromosome. It is only called a sister when it is attached. But when it becomes independent on its own, it now becomes its own chromosome. It can later on, during an S phase, regrow an identical copy of itself, and now it will become two sisters and a full chromosome. Okay, so this is still a full chromosome, and that's all you really need. You don't have to have both copies. These two copies are in case it wants to divide one day. All right, so here we are back up here in anaphase. So now we're going to go into telophase. So it's going to start pinching in. You get two new nuclei starting to form, okay, in both cells. So you got the first one going through it and the second one going through it. So this has its now chromosomes, and the pinching in is called cleavage furrow is forming. And then once cytokinesis completely finishes, so this is T2 and cytokinesis, you're going to get 4 brand new cells. These two will be identical, and these two will be identical, but this group will be completely unique to this group. All right, if they're going to be sperm cells, they continue on to get processed to get all their parts that they need. And we've already talked about if these were egg cells, really and truly, only one of them would be big. The other 3 would be really small because they're going to give all of their cytoplasm to one of those cells. So in egg cells, only one becomes a true oocyte, and the other 3 become polar bodies. Okay, so that's all right now for meiosis. We will, in this next unit, start talking about these genes that are carried on these chromosomes and how these genes interact and form all the characteristics, and the genotype determines the phenotype. But this is how these cells get made and how each organism can turn around and make a new cell that's half of their DNA and combine it with another new cell that's half of somebody else's DNA and create a brand new unique organism. Now, if we were bacteria, we would just do mitosis and divide and make a new one of us, and that's how our reproduction would happen. And that's just called binary fission, because binary fission is you divide and make an identical copy of yourself. Humans are very unique, and we don't do this. So we go through this process so that we get really good, unique, new genetic combinations of human beings.